Welcome back to another episode of the Iowa Beef Collective. I am your host, Patrick Wall. We are back again here at the Hansen Center, interviewing the guests that are on our program here at the Genetic Symposium. And with me for this episode is Jessica Peterson from University of Nebraska-Lincoln. And we're going to talk about a topic that's pretty near and dear to me, and that's genetic defects. In a previous career move, I was in part of the discovery of one of the defects that we test for today. Your talk this afternoon was what does a producer experience if they see something on the farm that this don't look right? What is the process that we go through? What do they go through? And kind of how the industry has developed, for lack of a better way to put it, a panel of defects that we test for today. So, Jessica, introduce yourself, talk about your career at UNL and what you do in the genetic defect world, and kind of how your talk here at the Genetic Symposium would benefit either a commercial or seed stock producer, either one.
Sure. So I'm Jessica Peterson. I'm an associate professor at the University of Nebraska-Lincoln. I've been there for almost 12 years, which is hard to believe. And I'm fortunate that the university already had a great team that was working to identify genetic defects in beef cattle. And so I'm a genomicist. So I bring in the expertise of doing the whole genome sequencing and identifying the mutations that might contribute to these defects.
So one of the first steps, in the process was I picked up the phone and called Dr. Stefan. Mm-hmm. And he was kind of the guy that researched what was going on, what the defect was. And then the other person that I'm sure most people are familiar with is Dr. Beaver. Right. He was at the University of Illinois at the time. I believe he's at Tennessee now. And so talk about, when a anomaly comes in, what is the process that the lab does?
Sure. So I don't see any of those usually until Dr. Stefan or another pathologist really has evaluated and ruled out things that are not genetic. And so most of the deformities or the defects that come to him, he can explain either through a bacterial infection, a virus, or some sort of deficiency or toxin. And so it's actually a small percentage of those that actually come to my lab. So when they do, often we bank DNA until we have compiled enough evidence that convinces us this really might be something genetic. And it's only then when we start to do the sequencing or the analyses to try to find out what mutation it might be.
So the anomaly, usually that's a phenotype, some sort of, whether it's lethal, which means the calf doesn't live, or a lot of the ones that maybe we're working on more recently, maybe they're not all lethal. The initial ones I think were, and that's kind of where it started. So, is the process the same whether the defect is lethal or whether it's not?
It really is, because it's kind of a game of statistics. We're trying to evaluate all the DNA in the genome and say, what do these calves that have this lethal phenotype or some other phenotype we're interested in, what do they have that nobody else has? So we can explain where that came from.
Okay. And I think, part of it is for the purebreds is pedigree analysis. If a defect pops up, there had to be an original mutation. And that has to show up if it's a double recessive allele. It has to show up twice in the pedigree.
Right.
So Dr. Stefan and Dr. Beaver do some of that work. If a commercial or purebred producer, has, let's say, a set of calves that all have the same the same phenotype problem. What do they do? What sample do they collect? Do they need an ear notch? Do they need the whole animal? What's the process?
Yeah, great question. So first of all, take as many photos and videos as you can. Often, Dr. Stefan or Dr. Beaver will get those photos, and they have so much experience in this that they can say, oh, this might be XYZ. And they can get on the phone with them and they can say, hey, I'd like you to send in the whole calf, or I'd like to get these samples from the particular calf. So they can really help direct that person on what samples will be important. From my side, when we go after the genetics, having a DNA sample is obviously critical. That can be really any tissue. Cut off a chunk of the ear is one that works really well. A TSU if the calf is living, or a blood sample. The purple top tubes are what we need for that. But in addition to DNA from that calf, it's critical to get DNA from its parents. Siblings, normal siblings are great because we have a good comparison then of what normal versus abnormal is going to be. So gathering all that information on those families is important.
And I think that, you mentioned the pictures and the videos. Those are important for a number of reasons, because I think once it is decided that, yes, there might be a genetic component to this, we need a way to, for lack of a better way to put it, promote that this phenotype is out there. And those pictures and videos are sent to the industry. And, with social media, we can get to a lot of people very quickly. And there will be breeders or producers that look at that and go, oh, I have seen that. And I think that's how a lot of some of the older defects that we've had for quite a while were discovered. One breeder might have said, hey, I lined up this genetic base and we're seeing this. But once the videos and pictures get out, there are probably a lot of other producers that have done a similar breeding program that go, oh boy, I've seen that too. And that's how we isolate the gene and discover the defect and move the industry forward and test for it. I guess talk about the process. Once we have decided, yes, it's a genetic component, we've got the sample, and I think I left this part out. If it's a lethal defect, it's very important we get that calf frozen.
Yep.
We can't, leave them lay out there, and the DNA is not the same if we wait a week. So we've got to get the tissue sample frozen. So that portion of a lethal defect has to happen fairly quickly. But once we've got samples coming in, talk about the development of the test from your chair. Like, what happens in your lab?
It's not as easy as it sometimes sounds. It's not a half-hour CSI show. But the first thing we would do is isolate DNA. We'd probably go ahead, even if we're pretty confident, and confirm parentage of that calf, especially if it could be a herd bull, there's multiple sires. We want to confirm parentage before we're going to spend any more money to sequence those animals. But it's incredibly affordable now to do whole genome sequencing. And so that's the path we usually take, is we will whole genome sequence, depends on how many cases we have, but 4 or 5 affected calves and their parents, ideally. Sometimes we'll sequence normal siblings, but we benefit now in that when we then get the sequence, we'll back up. We get the sequence, we align it to the reference genome. So that gives us an idea of how those animals differ from that reference and from one another. From that genome tells us where genes are, and particular positions of those genes are more important than others. So if we find variation in genes that might screw up how that protein works, those become of more interest. And then we can ask questions about, what do we know about that gene? Is that gene known to have a function in whatever this phenotype is that we're studying? So it's a series of filters. What do these calves have that the other ones don't? Then we look to public databases. What do those calves have that aren't found in other individuals? Because that helps confirm that it is unique to those. And then we ask questions about the gene. Is that gene, does it make sense? And can we demonstrate that the function of that gene is actually changed in those affected calves?
And I think it's, important for producers to understand that not every defect is the same. It's not always an autosomal double recessive causes a lethal.
Right.
We've got a bunch of them out there that aren't lethal, and some of them aren't autosomal only isolated to one spot either. Those can be a lot more complex to discover. I was part of one that one of the first ones that was incomplete penetrance. We didn't see the same phenotype in the same genotype. And it turned out that it was on the same chromosome as another defect. and that defect impacted the phenotype of this one. Sure. And so it is, I would like to reiterate that it's not easy to get these things isolated. You mentioned parents. We want to verify parentage. What if the bull that's the sire of this calf was an AI sire who's no longer with us? Can we use semen to get the genome sequence that you're after?
Absolutely. And actually, I prefer to use semen because sometimes mutations happen in his sperm cells that he might not have in the rest of his body. But that semen is what's going on to create the calves that have the problem. And so that's more reflective of what he is actually donating, if you will, to his calves.
Interesting. I just learned something. So I guess we've got a test developed. Talk about the commercialization of, does it automatically go on a SNP chip, or, maybe it does, maybe it doesn't. We've had SNP chips for quite a while now, and they have fairly wide coverage. But what are the chances that we find a defect that's not on a chip that we already have that is commercialized? Does that process differ? What do we do there?
Yeah. So the chance that we find a novel defect that's already on the chip is almost zero. And that's because those chips are built to assay known variation in the genome. So we can sometimes use that information if we have that on those animals to help narrow into the region, but we're not going to be able to use that as our commercial test. So often the genotyping company, whomever it might be, we share with them the information about the mutation that we discovered. And usually if they want to try to get it up and running very quickly, they have to have what they call a standalone test. So it's a specific assay that only is going to look at that particular mutation. The challenge with SNP chips is that they order them in big batches, and they can't easily add markers to them as they're identified. And so sometimes there's a lag time, and they'll be doing these standalone tests before they can put it potentially on a SNP chip.
So long story longer, just because we've discovered the defect and we know where it's at and we know the inheritance and we know all of that, that doesn't mean that we've got a commercial test that's just ready and we send it out.
No.
And I think that's important because, we're not done with genetic defects. We never will be.
Right.
And I also think it's important for the industry to understand that a bull has to be good enough to use twice before we'll ever see this, or a cow has to be flushed multiple times. So we're not going to see this in your typical run-of-the-mill, average commercial animal. Talk about your experience with the ones that you've worked on. What is the time frame from when it initially started to it's off my desk, it's done, it's commercialized?
Yeah, that varies. We've had one great success story, MD and Hereford cattle, where the time from when we sent the samples out to sequence to when it was published was within 8 months. And then that test was available very quickly soon after. That's unusual. So it's usually more in the terms of a couple years, because beyond the step to identify the actual mutation, there's a lot of due diligence we need to do to make sure we're correct. Before that, we send that to a genotyping company who's going to tell people you might not want to breed to this bull, or you might want to cull that animal. And so we do a lot of validation by genotyping a lot of animals to make sure it holds up. We also do a lot of validation to demonstrate that gene function actually is affected by that mutation in a way that makes sense to explain the phenotype we're seeing. So that takes time. And then the genotyping companies do that. They genotype things every day, but it also takes them time to get their assay going and validated so they know that they're giving people correct results to their test.
Good stuff. I think we'll kind of close with, I want, whether you're a commercial or a seed stock producer, either one, I think there is I'll call it a problem in the industry that they think, oh my gosh, if I get one of these, I can't tell anybody because, gosh, they might find out it was discovered at my place. And that fear needs to be gotten rid of. because the quicker that a producer will report an anomaly, whether it turns out to be genetic or not, is critical to how fast that process can work. I know in the one that I was involved in, we published pictures in a magazine asking for samples. Most of the samples I got were not from the breed we discovered it in.
Huh.
So I think, there's a lot of misconceptions out there that, the breed's going to come down on you or make an example of you. And there's nothing to that. If you visited with anyone within the industry, Dr. Beaver and Stefan and all of those that have come along the way, they want to find the answer as bad as the producer should want to know it as well. So any parting words of wisdom for the audience? I know the genetic defect stuff is I think one that they'll probably listen to on a podcast because they sure maybe don't want to admit they've ever seen anything that's an anomaly on their own farm. So hopefully we have educated our audience. But what's next in the genetic defect circle? Are we going to, you mentioned 8 months to 2 years. Are we going to shorten that time? Are we going to use new methods to make it easier to discover these things? Or has the science really come so far that I think we've got it where we want it?
I'm hopeful we're going to expedite that timeline. And one of the main reasons is because as we go through all of these, we're building up a wealth of information on what a normal animal looks like, or especially animals without the new thing that comes along. And so our ability to filter out non-causative variation in the genome is growing exponentially. Mm-hmm. That doesn't mean that they're all going to be fast. And there's a lot of types of mutations that are more difficult to identify than just a single base mutation.
Right.
And so some of those obviously will take different technologies and different approaches.
And I think, you talked about whole genome sequencing. We're genome sequencing livestock every day.
Yep.
And, we think back to the Dolly days, the first sheep that was ever done. I mean, that made the world news. Mm-hmm. So the industry's come a long ways. And I think, for good reason, we can really get rid of these problem genes much faster than we ever could. So I appreciate you joining us to describe to producers how that process works and how they can be a part of it. But thanks for coming on here. I know some folks enjoyed your talk downstairs and said we need to get her on the podcast so she can explain what that process is. So thanks for joining us. And always, the audience here at home, we appreciate you following along. Find us on the Iowa Beef Center Facebook page. We're on YouTube, Spotify. You'll find us on the socials. Appreciate you joining us. This institution is an equal opportunity provider. For the full non-discrimination statement or accommodation inquiries, go to www.extension.iastate.edu/legal.